Hello everyone, this is going to be chapter 2 of the chemistry of microbiology. Hopefully you don't hear too much background noise. I am not recording as late as I typically do, and so sometimes you'll hear like an ice cream truck or the occasional motorcycle driver, but hopefully it's not too bad. So this chapter we're going to talk about biochemistry and how it relates to microbiology and why it's important, properties of matter, elements, etc. So hopefully everybody at least is familiar with atoms and that they make up just about everything. And matter is anything that takes up space and has mass. So the definition of an atom is the smallest chemical unit of matter. So if you take a piece of anything and you cut it in half and then cut it in half again and again and again and again, it's going to get eventually to the small enough portion that if you cut it in half even more, it no longer has that systemic function or property. It becomes subatomic particles. And so we will talk about those, at least not until we get to the composition of the atom, but for the most part, they all retain the properties of that atom before becoming those subatomic particles. So everybody should also be familiar with protons, neutrons, electrons, and the nucleus of an atom. So the electrons are negatively charged, the protons are positively charged, and the neutrons are uncharged. So the nucleus contains all the neutrons and protons, while the electrons orbit the nucleus of the atom, forming the electron clouds that we'll talk about shortly. But those two, the ones inside the nucleus, the protons and the neutrons, are going to be the only ones that contribute to the mass of the element. And here you can kind of see that diagram where you have the nucleus in the center with protons and neutrons and electrons orbiting on the outside. And these electrons are organized in shells or orbitals, and these shells contain, the innermost shell has 2. Every shell outside of that has the capacity to hold 8 electrons. The outermost shell is known as the valence electron shell. Now, in terms of structure, the element is defined as the the smallest amount of matter, in this case an atom, that contains or exhibits the properties of that element. So the atomic number is going to be equal to the number of protons inside of the nucleus. It's really important to note that if you change the number of protons in the nucleus, you are now changing the identity of the atom, and that's not something that we want to do. We want to make sure that it maintains its properties. You can change the electrons, changing the charge. You can change the neutrons and change the weight. But the protons has to always be the same if you want to consider it that same atom. The atomic mass is the mass of the protons, neutrons, and electrons, but for the most part, the mass of electrons is negligible. When you see, for example, carbon has a mass of 12.01, the 0.01 is not because of the mass of electrons, it's actually because of the mass of the relative abundance of its isotopes, and we'll talk about what isotopes are shortly if you don't remember what those are. But again, the mass of electrons is really, really negligible. We typically don't consider it. Table 2.1 describes the element symbols and properties in terms of their biological significance. Make sure that you review those. So isotopes are an element that has a different molecular weight. In this case, we're talking about the number of neutrons being different. And there are some isotopes that are actually radioactive and they'll decay as they as time goes on, and some of those subatomic particles start being removed from the unstable core. So here we see carbon, for example. So carbon typically has a mass of 12 AMUs or Daltons. And when you have carbon 13 and 14, those are isotopes that have an additional number of neutrons. And so we can actually use these for radiometric dating, so we can say how old something is based on the proportion of carbon 14 to carbon 12, because we know the half-life of carbon, so we know how long it takes for carbon to lose some of that radioactivity. In terms of the electron configuration, we know that chemicals behave similarly when they have similar properties in terms of their electron configuration, and this is given to us by the valence shell of electrons. As you can see here, you have the first shell, which is occupied by 2 electrons, and then every shell after that containing 8. This is known as the octet rule, and depending on how many valence or how many outermost unpaired electrons there are, that's going to determine the interactivity of these atoms. So here you can see hydrogen, lithium, sodium, potassium on the left-hand side, so all of those are going to have pretty similar properties. On the other hand, all the way on the opposite side, you have helium, neon, argon, and those noble gases that are going to be very non-reactive. Everything in between is going to be according to those columns and the properties of that family. Valence again means the capacity of, or the combining capacity of every atom. So whenever you have a charge of +2, it means that you have an additional 2 electrons in terms of the number on top. And then you have things like oxygen that have a charge of -2. This typically means that it's missing 2 of its electrons. And so whenever you have a +2 and a -2, they're going to want to come together to stabilize that octet, meaning that that shell is full. They do so by forming chemical bonds, and these chemical bonds are going to have different properties based on which atoms are making them up. And these molecules are essentially just 2 or more atoms combined. A compound is a molecule that has more than one type of element. Molecules can be the same type of element. We have different types of bonds. The first that we're going to discuss is the covalent bond. Most of the things we have in our bodies are going to be covalently bound, as well as other organisms. They typically are going to have that covalent bonding, mainly because we are carbon, hydrogen, and oxygen in about 96% of most of the living tissues that we have. And these bonds are going to form because of the electronegativity, or the difference in affinity for electrons. So things that are more electronegative are going to attract electrons a lot more than things that are less electronegative. And this is what we know as the pull of electrons. So in terms of electronegativity, they do have assigned values. For example, lithium, sodium, potassium being not very electronegative, and then the inert gases are highly electronegative. The most electronegative element in this case here is fluorine with an electronegativity of 4. So if we were to look at an atom, for example, like carbon, if you compare to the pull of carbon to hydrogen, carbon has a slightly stronger pull of electrons, but because typically a carbon is surrounded by hydrogen atoms, it neutralizes that difference in pull, making atoms that are predominantly carbon and hydrogen non-polar. And so we'll talk about what polarity is in a little bit as well. So nonpolar covalent bonds are bonds that form because electrons are pretty even when it comes to their distribution. So again, most of the pull of these atoms is even across the board, and so therefore they're not going to have any poles. And poles are essentially areas where you have a high concentration of electrons or a high concentration of protons. So here we have, for example, hydrogen gas, which is H2. As you can see, the electrons are pulled evenly in each direction by each hydrogen molecule, therefore no pull is exhibited. The same is true for oxygen. They're going to be pulling equally in either direction, and they're going to form no pulls. Like we talked a little bit earlier, carbon and hydrogen do have a slight difference in electronegativity, but their pull is just about equal when you are surrounded one of them with the other. So in this case, there's really no true area where you have an excess of these electrons. They're being shared or pulled evenly in most other directions. Now, when it comes to polar covalent bonds are where you have unequal sharing of electrons. And this is important because it's going to leave a cluster of electrons somewhere making that side of the molecule negative. And because you don't have electrons everywhere across the molecule, where your protons are kind of left exposed, that's where you're going to develop that positive pull. So here we have water, for example. Water is a very interesting molecule. It is a bent geometry, and it does so because of something known as the valence electron repulsion theory. So what it is if you can look at the molecule or figure A, you see that there are 6 electrons clustered on top of this molecule. So what those are doing is they're creating this cloud of electrons, and that cloud of electrons is pushing the hydrogen atoms downward, therefore causing the molecular geometry of this water molecule to be bent. And so that, in this case, because you have that nice little cluster of electrons up top, that's going to create a negative pull, and the protons are going to be exposed now at the bottom because the oxygen is pulling them much harder than the hydrogen is, causing a positive pull. So this is a perfect example of a partial positive, partial negative molecule, or a polar molecule. Now, ionic bonds are the weakest type of bond. We really don't have many of these in the body other than in some proteins, mainly because we're a lot water, and if wherever you have water, it's going to typically disassociate these ionic bonds. They're not very strong at all. But when they do disassociate, you have the formation of ions. More specifically, you're going to have cations, which are positive, and anions, which are negative. And so these ionic bonds are typically going to be dissolved or disassociated into things known as electrolytes, and typically are going to come from things like salts. So here you have sodium and chlorine, which together make sodium chloride. So in this case, these electrons between these two atoms are going to be transferred. Now in covalent bonding, those electrons are never transferred, they are always shared. The electrons are pulled by both atoms, and this is what's going to create the bond. That's the intramolecular force. Here, the electrons are transferred, so they are given or taken from one atom and given to the other. In this case, that leaves for no pull in between these atoms, therefore they can very easily disassociate. However, one of the benefits of this type of bond is that those atoms are quite stable, at least until they come into contact with water, where they become ionized. But at least here in their ground normal state, there's really nothing holding them together, other than like the lattice structure of these of the crystals that will form eventually. Here we see that occur with sodium and chlorine. As you can see, when you add water, it'll disassociate that bond, breaking it completely. Now there's nothing holding the sodium and the chlorine together. Hydrogen bonds are another type of weak bond, but they're kind of strong in numbers. So think about a tiny little thread. So if you take one little strand of a thread and you wrap it around your arms and then you tell somebody to escape, they could probably break it quite easily. But let's say that you surround them with 100,000 of these individual threads. Now that's going to make it a lot more difficult. So hydrogen bonds are kind of like that. When you have just one of them, they're pretty easy to break. But when you have hundreds of thousands of them, it becomes a quite significant bond. And this is the case for things like DNA. So DNA, the backbone of DNA is held together by covalent bonds, but in between the two strands of DNA, that's where you have hydrogen bonds. So that's the entirety of your DNA molecule is held together by these hydrogen bonds, or at least the double helical structure. Here you can see cytosine and guanine, which are purine and a pyrimidine that are responsible for forming your genetic code. As you can see, there are bonds between the hydrogen and the oxygen and the hydrogen and the nitrogen of these opposing molecules. Again, these are weak bonds individually, could be broken quite easily by any type of enzyme, including DNA helicase, which is one of the ones that gets the process started. But when you have thousands of them, they're quite significant. Now, chemical reactions are quite interesting because they're one of the ways that things can change throughout the body, and we're talking about building and breaking specifically. And they involve having reactants and products, and biochemistry is a study of looking at how these reactions work. You have different types of reactions, the first of which is a synthesis reaction, and like its name implies, it is there to create larger, more complex molecules. It is an endothermic reaction. It does require energy. Typically, this energy is in the form of ATP or other sources of energy, but they are also known as condensation or dehydration reactions. This collectively refers to the anabolism of an organism, so everything it builds in its lifetime. So here we have an example with a simple small molecule. On one of the end, it has a functional group of the OH, which is a hydroxyl group. On the other end, there is an H. And so whenever these two molecules come together and they are meant to be built, an input of energy and an enzyme are going to take these two molecules, remove the H and the OH, form water, but at the same time create a connection in between the two molecules that were existent before. And so that forms a bond. When you do this to carbohydrates, it's known as a glycosidic linkage. When you do this to proteins, it's called a peptide bond. When you do this in lipids and fats, it's called an ester bond. And when you do it in nucleic acid to form the backbone of DNA, for example, it's called a phosphodiester bond. And so there's quite a bit of diverse bonds that occur this way. And decomposition is the opposite. So where one is a condensation reaction or a dehydration reaction, then you also have the hydrolysis reactions. So hydrolysis reactions break down those bonds, again, with help usually of an enzyme, and they are responsible for the collective catabolism of an organism or the total breakdown of things. They are exothermic in nature, so as well as splitting that bond, they're also releasing the energy that bond contained. This could be in the form of heat or other forms of energy, of course. So here we have that model, same one, 3 small little blocks. Whenever you introduce water, an enzyme, and a little bit of energy to get that reaction started, you have a payoff in the form of energy. And you have those 2 molecules that form. So these reactions happen a lot. So some of them are building and some of them are breaking. In reaction, this refers to the metabolism of an organism, the sum of the breakdown and building of molecules and whatever they offset. And water is really important for this. A lot of these organisms require water. Water is the stuff of life. It is one of the things that can force reactions just because of the interaction that's happening within the molecule, the flexible hydrogen bonds, and a lot of the different properties like the specific heat and so on and so forth. Right? But it is an excellent solvent. It can dissolve just about anything given enough time because of its dipole moment, meaning that it has that positive side and negative side that can just destroy just about anything. It also generates surface tension, which is the attraction of water molecules to themselves. Now this is kind of interesting because every summer I seem to have ants that like to get into my house, and they just like to ruin my pantry, and I hate them. And so whenever I collect them, sometimes I throw them in a big bowl, and I see them trying to float to the surface, and they do successfully. That is until I introduce a drop of soap, and that drop of soap, and I know I'm laughing and it's horrible, but that little drop of soap actually breaks the hydrogen bonds at the surface, and it doesn't allow for the surface tension to form, so they'll eventually sink and drown. And I know that sounds really terrible, but you know, in this case, I get rid of the ants because they ruin the stuff in my pantry. And I know it's a pest, but I try to ward them off with other things. But you know, sometimes they just bug me that much. Don't try this at home, by the way. But anyway, yeah, there's a lot of different things that happen with water. It's a really interesting, important molecule. So here we have a water strider. Lives on the surface of the water. It's typically a predator, and it'll run along the surface of the water whenever it detects like little ripples of something that fell in, and they'll go and pierce it and then suck up the juicy intestines or whatever it is that they have on the inside. Now, when it comes to water, it's really important because water is pure water is a neutral substance. It has a pH of 7, meaning that it has equal numbers of hydroxyl, OH, and hydrogen, or H plus, ions. So when they go in water, one of them, of course, is going to form the hydroxyl. The other one is going to form something known as hydronium. So hydronium is the acidic part, and the hydroxyl is the basic part. And so there's always a delicate balance, but when you introduce more of hydrogen ions to the solution or more OH into the solution, it's going to change something that we call the pH, or the power of hydrogen. And this determines a lot of different things, including the ranges of conditions that some organisms can tolerate, to how reactive a solution is going to be when you pour it into something that has a different pH. And so in our body, we have lots of buffers that can resist these changes. That's why we're able to consume things like wine and soda and lemon, tomato juice, because those are really, really acidic foods. But those buffers help us resist those changes in pH. And we'll talk about those a little bit later in the course as well. So here we have a strong acid and a strong base. When you place them in water, they will disassociate. So for example, we have hydrochloric acid, which is HCl. If you were to put that in water, that is a strong acid. The H will disassociate, forming H plus ions, and the chlorine will also dissociate, forming Cl minus ions. So HCl, when it's in its liquid form, it'll stay together, but once you put it in water, it'll dissociate into those smaller ions. So this is going to change the pH of the solution, making it more acidic. On the other hand, let's say that you have sodium hydroxide, NaOH. So the Na is going to disassociate into positive cations, and the OH is going to stick together but disassociate as an OH group or a hydroxyl group. So this is going to change the pH to become more basic. In terms of the scale, things that are highly acidic are going to have a pH closer to 0. If they are more basic or more alkaline, they're going to have a pH closer to 14. So humans, for the most part, we have ranges between 6.5 to 7.5 in terms of our pH. That's where most of our functioning cells do their work. Like I mentioned earlier, buffers are things that will prevent drastic changes in pH, and again, it's because we need to maintain homeostasis, because acids could be particularly damaging. And if you remember from an anatomy course, if you've taken an anatomy course and um, remember how CO2 is removed, CO2 is removed by the respiratory system through the use of bicarbonates in our bloodstream. And so if the pH of your blood gets too acidic, you start breaking down some of those bicarbonates and you are unable to remove carbon dioxide waste from your system. And so although these buffers are there to protect from things like that, they're there to prevent some of these processes from going haywire or becoming interrupted. A lot of microbes definitely prefer ranges of pH between 6.5 and 8.5. So when we grow them in medium, sometimes they have to be pH controlled. But we'll talk about that when we get to the different types of media. Additionally, I forgot to mention that some microbes can definitely influence the environments in which they live in. For example, some fungi, they release something known as acid phosphatase. And acid phosphatase is a chemical substance that is meant to degrade phosphate ions from the environment so that the fungus can uptake them. And so they're quite interesting. A lot of others have different, more interesting features and activities, but I just thought that one was particularly interesting to me. And so salts, like we said earlier, can also dissociate. That's mainly where you're going to have most of your ions. And yeah, they become electrolytes, making it possible to transfer things like electrical charges, in this case from electrons, but also make really important parts for enzymes or ions that enzymes are going to require for proper functioning. Now, in terms of the functional groups, a lot of macromolecules are going to contain carbon, they're going to contain hydrogen, some of them are going to contain hydroxyl groups, which are the OHs, and we call these molecules macromolecules because they're typically large, they contain a bunch of groups, and we have in this case 4 classes that we'll talk about, which are your lipids, carbohydrates, proteins, and nucleic acids. Now monomers, these are the basic units, these are like the tiny little individual pieces. So let's say that you wanted to build a Lego house, the smallest unit in this case would be the Lego brick. And so these Lego bricks are going to assemble together to create your Lego house. So these are just some of the functional groups. Make sure that you familiarize yourself with some of these as we will be talking and using them extensively throughout the course. We'll start with the first type of molecule, which is the lipid. So lipids are fats, oils, and waxes. They're not composed of regular subunits. This is one of the things about lipids that makes them unique. You will typically have several different types. You'll have steroids, which have ring shapes. Then you'll have triglycerides, which do form similarly to some of the other compounds. They'll use ester bonds and dehydration reactions to form them. But those are your triglycerides, really, really common. Phospholipids are going to have a choline group as well as 2 fatty acid tails, which give them their traditional shape, and we'll take a look at some of those in a little bit. And then, of course, your waxes and steroids. So these right here are your triglycerides. They're composed of a glycerol molecule and 3 fatty acid chains, making them the triglyceride shape. Of course, these are happening or forming due to a dehydration reaction. And it's really interesting how you can see the differences in their shape. So some of them have the squigglies that are fairly straight. Those are known as saturated fatty acids. So saturated fatty acids are acids that have complete saturation when it comes to the number of hydrogens that they can have. So as you can see, every carbon is bound to 3 different hydrogens, and that is complete saturation. Now, when you have a carbon that doesn't have 3, that forms a little kink, as you can see in the figure B. So that little kink gives it an irregular shape. Now, there's some interesting things that can happen with lipids that are like this. One of the most important characteristics is that they have different properties. So saturated fatty acids, these are the types of fatty acids that will form when you get animal fat, for example. So think about the last time you cooked bacon, if you've ever cooked bacon, or you've had pozole or menudo or any type of like fatty soup. When you put it in the fridge or when you cool it down, it starts to form solid shapes. So in the case of soup or menudo or stew, you can have these little circles form on top of your soup when you refrigerate it. In the case of bacon fat, when the pan cools, you have this layer of white fat on top of it. That is saturated fat. Lard is an example of that. Crisco is an example of that, the vegetable saturated fat. That's known as a hydrogenated vegetable oil. And all you do is you take an unsaturated fat and you saturate it. That's how we get Crisco. It's pretty bad, but I'm not gonna lie, it's probably really, really delicious in pastries and stuff like that. But anyway, just some properties of those two that I found interesting. So this is a phospholipid. So phospholipids have a choline group, a phosphate group, and two fatty acid chains. That's what the organic group is. It's a choline. And so these are what's gonna compose the lipid bilayer of all of your cells. So this is the molecule responsible for keeping things out of the cell and into the cell or inside the cell. Waxes are really important. They're completely insoluble in water. They're mainly long fatty acid chains that have ester bonds. They're covalently bound, and some very, very primitive lineages of bacteria, I believe, use wax as their main storage for energy. Lastly, we have steroids. So steroids are these ring-shaped molecules. Eukaryotic organisms specifically embed these in the cell membrane just to make it a little bit more fluid so that it moves around a little bit more freely, more organically. And when you don't have this, it becomes a little bit more difficult for these to move. It's easy for these membranes to rupture if they don't have enough of it. And at the same time, they can make it hard for things to permeate through them. The cholesterol molecule that we have here, or the derivative storage from cholesterol, in this case could be testosterone, estrogen, or whatever it is, it's quite bulky. So when you have it within the membrane, it's difficult for things to pass through it, and so it's going to make it a little bit more impermeable to things. Next, we have our carbohydrates. They're mainly carbon, hydrogen, and oxygen. The simplest, most interesting one that we're going to be dealing with is going to be glucose, which is C6H12O6. It's typically a mechanism of storing energy, but also we can use it quite easily. It's ready to use. It can circulate through your bloodstream, and a lot of organisms are able to synthesize that. You have it in cell walls, you have it in just about everywhere. You can even have it on the surface of the cell as a messaging or identity label, just to make sure that cells don't attack it. For cell recognition, they're super important. We have our monosaccharides, disaccharides, and polysaccharides, and as their name implies, mono, of course, meaning one, disaccharides meaning two, and polysaccharides meaning multiple. Here we have an example of a simple glucose molecule on top. So these are two glucose molecules, or have the composition of glucose molecules, but they have a difference, the alpha and the beta configuration. So why does it even matter that there's an alpha and the beta? So if you look at carbon number 1, you see that the H and the OH are kind of flipped. And does this make a difference? And if you don't know if it does make a difference, you have to think about the fundamental principle of enzymes and their substrates that we talk about in general biology. So if the enzyme doesn't have the correct shape, the substrate cannot be broken down. And so in this case, let's say that you are a human and you are eating a potato and you're eating rice and you're eating corn. All those things are made from starch. So starch has monomers of the alpha glucose configuration. So you can take that, you could eat it all day. But now let's say that you decide you want to go outside and eat some grass because you think you are a cow or deer or I don't know what animal you think you are, or an insect, and you start eating this grass. Now you do not have the proper enzymes within you or the number of stomachs, like if you were a cow or something like that, to break down the beta configuration of glucose. So alpha configuration can form something known as an alpha-1,4 glycosidic linkage, but the beta configuration forms a beta-1,4 glycosidic linkage. You do not have the proper enzymes to consume the beta forms, only the alpha forms. And so if you're wearing a shirt that's made of cotton right now, for example, that's something that you cannot eat. I mean, you could technically eat whatever you want, right? It doesn't mean that your body's going to digest it. And so similarly, organisms that are not equipped to consume the alpha-1,4 versions are probably not going to do so. But in this case, just about everything eats the alpha-1,4. But that beta-1,4 makes some interesting stuff. For example, it can make up the walls of cellulose of most plants. So when you're eating plants like corn and you thought you chewed it really well, and then later on you realize that you didn't chew it so well, that stuff is dietary fiber, so you cannot digest it, and so therefore it just goes through your digestive system intact. And so that's kind of the difference between the alpha and the beta. And I know it's a big explanation. I'm not entirely sure if this is covered in depth in your text, but I do ask you to know that. And then you have your N-acetylglucosamine. So N-acetylglucosamine, and we're going to call it NAG for short, is a glucose molecule that has an acetyl group attached to it. And so this is important because wherever you have amine groups or acetyl groups or anything that's not something like a simple OH group, like a carbonyl, then you have the potential for bonding. And so when you mix some of these molecules together, you are removing water to form in this case, glycosidic linkage, but then that allows you to create more complex molecules. And so when we talk about some of these disaccharides and polysaccharides, they can form these interesting shapes. So the alpha 1,4, alpha 1,6, they can form helices, they can form branched patterns. But when you have those beta 1,4s, these are going to be the ones that are going to allow you to form sheets. So sheets are really important for the cell walls of some organisms, including plants. But when you combine things like NAG and other things, you're able to make some interesting patterns, and that's kind of where the next thing comes in. So proteins are mainly carbon, hydrogen, oxygen, nitrogen, and sulfur. They have so many different functions. I know these have 5, but for the most part, structure, it's going to be something that is not necessarily too in-depth covered, but enzymatic catalysis is definitely going to be important. Transportation, defense, offense, we'll talk about that stuff in the chapter 4 immunity, but the regulation of certain processes, inhibitory pathways, and all that stuff, those are all things that are controlled by proteins. Now, amino acids, there only are 21 of them that make up just about everything that you've ever known, everything that even you haven't known. So all proteins are derived from these 21. They're unique three-dimensional shapes. All the different antibodies are created by every DNA sequence. Those are all made by proteins. So proteins are covalently bound, so they tend to be quite strong, and they exhibit different kinds of interactions. So on the left, you have the amino group. When you insert it in water, it becomes ionized. It becomes an NH3 group. Then you have the central carbon, which is in the middle. One of them has a hydrogen. The other has a variable side group, and this is known as the R group. These could be polar, they could be nonpolar, they could be acidic, they could be basic, they could be electrically charged. They could be a bunch of things, and so that's kind of where the diversity is. And so one of the things that is important to note about these amino acids is that they will interact with each other. So here you have some that have some ionic bridges, like the sulfur bridges, some of them that have another amine group attached to them. So they're going to interact in interesting ways. For example, if you have one that is polar and one of them that is non-polar, they're going to repel each other, whereas two polar ones are going to attract each other. And so this is what's going to determine the overall shape of this protein as it forms into its three-dimensional shape. You can also have some of these that are mirror images of each other. These are known as stereoisomers. Isomers because they're made of the same molecules, stereo because they're kind of like right compared to the left orientation of these molecules. They are also held together by dehydration reactions or in this case, a condensation reaction. So these right here are combining the OH and the H groups to form a bridge in between them known as the peptide bond. The peptide bond you can see highlighted in pink on the right side of the equation. So when these peptide bonds end up forming, you now have these long chains, and then based on the interactions of the R groups, you're going to have either an interesting two-dimensional or three-dimensional shape. Once those three-dimensional shapes form, they can come together in bunches and aggregate and create what's known as quaternary proteins. And these quaternary proteins are even more complex, and they are a combination of three-dimensional, two-dimensional, and chains of amino acids. As you can see, they're stabilized by different things. So for example, a primary structure of a protein is determined by the sequence of the peptide bonds. The secondary, or the alpha helix or the beta pleated sheet arrangement, is stabilized by hydrogen bonds. And the tertiary structure of a protein is determined by the interaction amongst the R groups. And so there's different things that stabilize these and help form them, but you have to remember that the primary thing is that they're scaffolded. So if the primary structure fails, so does the secondary and tertiary. If the secondary structure fails, so does the tertiary. And so they have to build upon each other to have a good functioning protein. Now, the last one we're going to cover is deoxyribonucleic acids. These are the ones that help make DNA, and ribonucleic acids are the ones that make RNA. So this is the genetic material that's found in your cells, but they also have another important role. So these nucleotides, they're made of three things. They're made of a sugar, a phosphate, and a nitrogenous base. The sugar could be either ribose or deoxyribose. The sugar is typically a 5-carbon sugar known as a pentose. The phosphate group is going to be a phosphorus attached to a couple of oxygens. And then you have the nitrogenous bases, which are adenine, cytosine, guanine, and thymine. So here you have that very, very basic shape of them overall. You have your purines and your pyrimidines. And there at the bottom in figure B, you see the difference between the deoxyribose and the ribose. Ribose has an OH, deoxyribose only has an H in that position. So what that's going to mean is that that oxygen, which is highly electronegative, is not going to cause the same level of interaction as the H does. So DNA tends to be a lot more stable than RNA because of that difference in the structure of that sugar. Now, nucleic acids are really important. They are the ones that are going to hold together your DNA, or what's going to form your DNA. Again, those hydrogen bonds are the ones responsible for holding the two strands together. So there's 3 between the C and the G, 2 between the A and the T. And of course, that double-stranded is complementary and antiparallel. Hopefully you remember those terms from your general biology. Again, antiparallel meaning that these strands are going in opposite directions. So they're parallel, but one of them is facing up, the other one is facing downward. So kind of like you see in that figure right there. So if this would be the 5 to 3, the 3 to 5 would go the opposite direction. So nucleic acids, again, they carry the information in terms of your genetics to code for proteins. They can control different pathways, and they're also important for other biochemical processes. Table 2.5 gives you a comparison of DNA and RNA, what the differences are. But in terms of the nucleic acids, ATP is made from nucleic acids. So it's an adenine that's been modified to have 3 phosphates. So these 3 phosphates are going to be the source of energy for just about every type of metabolic pathway that we discuss. Whenever you form these bonds hold energy. The moment the bonds are broken, the energy is released. Now, ATP is not a long-term storage solution for energy. It's an unstable molecule. It's going to degrade, and every time it degrades, it's going to release energy to do chemical reactions. So here we have what it looks like. So typically when you have an adenosine triphosphate, it has the most energy once you split that last phosphate, which contains energy to do work. This is for just about any enzyme that your body has or cells have. You will form ADP, which is adenosine diphosphate. Once you split that one, you're going to have adenosine monophosphate. So there's a version of this called cyclic AMP that we'll talk about, as it is a very important part of communication for hormones and other things like that within the cell. We'll talk about that later. And then you also have just adenosine, which is the sugar and the adenine. And that is the end of this lecture. I will make sure to post another biochemistry lecture along with this so that you can take that supplemental material in case you need a refresher.